signal is emitted, it is there for anything in range to detect. However, only target cells are able to detect the signals. This is because they have a receptor protein either on their surface or inside them. The target cells will then respond. The receptor and signaling molecules fit together similar to a lock and key. In this case, the signaling molecule is a ligand, which is a molecule that specifically binds to another molecule. When this binding happens, the receptor protein will change shape. When it does this, it activates the receptor to communicate with molecules. If the change of shape doesn't activate the receptor immediately, it may cause receptor molecules to cluster up, which will eventually cause activation. There are two types of membrane receptors, intracellular and cell surface. The majority of receptors are on the plasma membrane of the cell. The three types of cell membrane receptors are G-protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. These receptors are responsible for impairments that cause common health defects. It is very hard for scientists to study these receptors because they are difficult to crystallize. Malfunctions of receptor tyrosine kinases are responsible for cancer diagnoses. Receptors inside the cell are called intracellular receptors. These receptors are found on the nucleus or cytoplasm of a cell. Signal molecules get to these receptors by traveling through the membrane. They must be small enough or hydrophobic enough. When they reach the receptor, it changes into a hormone receptor complex and can control whether genes are on and off. So what kind of molecules bind to each receptor? For cell surface receptors, polar molecules bind to them. They bind to receptor proteins and they don't enter the cell. Receptor proteins send information from outside the cell to inside. For intracellular receptors, nonpolar molecules bind to the receptors. For example, steroids. They pass the membrane through diffusion. After a signaling molecule binds with the receptor, the first step in the signal transduction pathway begins. At each step, information is passed to a new cell and carried to another. Also at each step, the information changes shape. This is called by phosphorylation. Signals can be amplified by secondary messengers. Cyclic AMP, as seen here, moves through the cytosol and moves signals quickly, as does Ca2+. Tyrosine kinase pathways also use diacylglycerol and inositol triphosphate as secondary messengers. So why do they change shape? That is due to phosphorylation. Protein phosphorylation is when a phosphate group is added to a protein which activates it and changes its shape. A protein kinase is what moves a phosphate group to the protein from ATP. In phosphorylation cascade, proteins are phosphorylated repeatedly and each activates the next. Dephosphorylation is the removal of a phosphate group. Protein phosphatase removes the phosphate groups.
The cell response occurs after transduction and reception. A signal transduction pathway leads to the regulation of one or more cellular activities. The response can occur in either the nucleus or the cytoplasm. So this is the nuclear response. And a nuclear response activates a specific gene by a signaling molecule and it alters the gene expression. This starts with reception. So the signaling molecule will attach to the receptor, and this causes phosphorylation cascade to occur and transduction. The last kinase from the sequence enters the nucleus, so this is the nucleus, and a transcription factor is activated. So this is when the inactive transcription factor goes to an activated transcription factor. A specific gene is activated, and the mRNAs direct the synthesis of another protein. So a cytoplasmic response is different because it regulates the activity of proteins and doesn't cause synthesis by activating gene expression. It opens or closes the ion channel in the plasma membrane or involves the activation of an enzyme. It begins with epinephrine binding to a G protein coupled receptor in reception. In transduction, relay molecules are activated. So all of these are different types of relay molecules. And finally, cellular response converts glucose to glucose 1-phosphate. Two types of cells can respond to the same signal in different ways because different cells turn on different genes and they have different collections of proteins. A cell response depends on the type of receptor proteins, relay proteins, and proteins needed to carry out the response. For example, this cell responds to a signal like this, a single response. A different cell with the same signal responds with branches leading to 2 responses. The G protein was shown in the cytoplasmic response because epinephrine binds to a G protein coupled receptor in reception. G proteins are made of many proteins that act as a switch in cells. They are involved in transmitting signals from outside the cell to its interior. G proteins can bind GTP and GDP. The G protein signaling pathway may be activated by a ligand binding to the G protein coupled receptor. This leads to cellular responses. In this example, CAMP is used as the second messenger in a G protein signaling pathway. So first, the first messenger binds to the G protein coupled receptor and activates it. Then the activated G protein coupled receptor binds to a G protein, which is then bound by GTP activating the G protein. That binds to adenyl cyclase and it becomes activated. The activated adenyl cyclase converts ATP to CAMP. And lastly, CAMP activates another protein, leading to cellular responses.